good afternoon, and good evening again. Let me start. My name is Elena Moreno. I'm the Senior Project Manager at the Global HIV Vaccine Enterprise for the International AIDS Society. It's my pleasure to open this webinar organized by the Global HIV Vaccine Enterprise. The event will follow in the steps of previous enterprise webinars that discuss experimental medicine research and will bring regulatory scientists and ethicists to discuss how to improve knowledge and capabilities of regulatory authorities and ethics review committees to assess experimental medicine clinical research in high and low and middle income countries. I will send you later the link in the chat in case you want to watch previous sessions on experimental medicine. It's my pleasure, and I would like to welcome all participants as well as our speakers and panelists who will be introduced to you shortly. Please note that all participants are muted. However, we encourage you to use your questions and answers. You can type them in the questions and answers box. They will be answered at the end of the session. We will start first with three presentations, followed by a panel discussion. And having said that, it's my pleasure now to introduce our chair, Cathy. She is a clinical psychologist and head of the HIV/AIDS Vaccines Ethics Group at the University of KwaZulu-Natal in South Africa. HAVEC is involved in conceptual and empirical research into ethical legal complexities in HIV prevention trials, and HAVEC is part of CASP project, a coalition to accelerate and support prevention research. It's my pleasure. Cathy, please, all yours. Thank you.
Thank you. Thank you so much, Elena. If I could just be put on video, I think it's just been stopped. But in the meantime, I'd just like to welcome everyone to the webinar and to say thank you so much for giving up this time just to look in a very dedicated way at this particular topic. And we are going to be spending this time hearing from presenters and panelists and attendees. Obviously, we hope you will speak to us as well about some of these issues, but it's on this important topic of how best to support national regulatory authorities, as well as research ethics committees, or IRBs as they're sometimes called, who are tasked with the ethics review of these trials. And as many of you will know, and with all kinds of research, review authorities need to be able to conduct efficient, but also high quality review. So in other words, review that's not delayed, but also doesn't sacrifice any quality. And this needs to be done across all settings. So we may look at LMICs today, but it really will have to be done in a variety of settings, not limited to LMICs. So we really just want to welcome you to this session. It hopes to dig a bit deeper into both the complexities and opportunities presented in these review processes in two settings, both the regulatory process and the research ethics committee process. And we're hoping to try to tease out some of the threats to efficient and high quality review. Think about what applicants could do, researcher applicants could do when they submit protocols, and what reviewers could do when they review them. And importantly, try to identify existing platforms, existing tools and resources that might facilitate this and support this process. Just to reiterate what Elena said, please feel free to put comments and questions into the chat, any questions or reactions. It's really wonderful to have you all with us. And at this point, I'm going to move on to the presentation section. The first presenter that we have with us is Eric Karikari Boateng. He is Director at the Center for Laboratory Services and Research at FDA Ghana. And Eric is going to do a presentation for us on a regulatory authority perspective. Over to you, Eric, and thanks very much.
Yeah, thank you, Cathy, and good afternoon to everyone. And as Cathy has already said, my topic for this afternoon is experimental medicine trial perspective from the regulatory authority. So this is going to be the outline of my presentation. The first is, how do we define experimental medicines, phases of clinical trials, which everybody is aware of, then regulation of clinical trials in sub-Saharan Africa, challenges facing NRAs and ECs in sub-Saharan Africa, and then the way forward. So how do we define experimental medicines? So we can be said scientifically as a product being tested or used as a reference in a clinical trial containing an active ingredient being tested or placebo being tested. It's used as a reference in a clinical trial. And this includes products with marketing authorization, a medicine, medical device, or procedure, or herbal medicine. Like this is the last point in the Ghana law, herbal medicines are also classified as medicine. So they're specific to the law in my country. And then the question, how do we define clinical trials? I mean, this is a basic definition of research that is conducted to determine whether an intervention is safe and effective for humans. And traditionally, there are 4 phases: phase 1, phase 2, phase 3, and phase 4. But as COVID taught us last year, we can know that now some of these phases overlap because adaptive trial is now becoming the in thing. So, like in a phase 1 trial, it typically involves 20 to 100 healthy volunteers. Normally, you don't see women of childbearing potential because at this point, most companies or most sponsors would not have conducted reproductive toxicology studies. And we should know that, like some oncology products too, it might involve patients. Patients will have to be used at this stage. The emphasis is on drug safety, and the main goal is to identify its safety and tolerability, to identify major side effects, metabolisms, and route excretion. You know, this lasts about a year, and about only 70% of drugs will pass this phase, and the remaining 30% will fail, and that will be the end of their development. And phase 2, that's the classical phase 2, typically involves 100 to 300 individuals who have the disease, who have the target disease. Normally it's designed to include a control arm, and randomization. Assignment is by random assignment. And patients receiving the drugs are normally compared to similar patients receiving a placebo or an active comparator. That's if there's an existing drug that has been approved in the jurisdiction where the study is taking place. And it provides a preliminary information on the efficacy of the drug and also additional information on safety. I mean, that you can determine the common short-term side effects. And this lasts for about 2 years. And normally the pass ratio is about 33% of the drugs. The remaining 67 will be assigned to the mortuary, research mortuary. Then we have clinical phase 3, and this typically involves large numbers, about 1,000 to 3,000. Then FRC is on safety and efficacy, and also effectiveness. And this is investigated to well-controlled studies, different population, different dosages, as well as uses with new drugs in combination with other existing drugs. And what the main objective is, we want to see whether the results that we saw from the proof of concept in phase 2 can be reproduced in multiple centers across multiple population. And normally about a third of this drug will make it from here and will apply for marketing authorization, which most likely all of them might not be successful. So, and then there's a conventional clinical phase 4 studies as post-market surveillance of drugs to continually assess the safety of the drug post-approval. And sometimes it can be a post-approval safety study. It can be a post-approval effectiveness study, and may include mainly incidence and severity of rare adverse reactions, cost-effectiveness analysis, comparative trials, and then quality of life studies. Adaptive clinical trials. As we all know, COVID, formerly regulators were not very comfortable with adaptive clinical trials. As I have been saying, it's now becoming the in thing. It's the use of accumulating data to decide how to modify aspects of a trial as it continues without undermining the validity and integrity of the trial. Examples of potential adaptation include stopping the trial, modifying the allocation ratio, re-estimating the sample size, and changing of the reference, as well as eligibility criteria. We all remember the recovery and then solidarity trial from the COVID. The most valid adaptive designs are those in which the opportunity to make adaptation is based on pre-specified decision rules that are fully documented in the protocol. And this is what regulators would like to see. Phase 1 to 3 of a clinical trial could be combined for implementation, and the cost of the trial will be seamlessly transition from one phase to the next. And that's what I said we saw a lot during the COVID-19 trials. So, how were clinical trials regulated before the Aurum era? Yeah, basically, the regulation of clinical trial on the continent was practically non-existent. So I think WHO did a survey and the year 2005 and realized that some 19 countries that had been targeted for clinical trials for this malaria, TB, and HIV, and they realized that basically capacity was non-existent on the continent with respect to ethical oversight and then regulatory oversight. So in December 2006, a decision was made to form the Aurum. It started as the African Vaccine Regulatory Forum. And this was a platform for NRS and independent ethics committee in Sub-Saharan Africa for clinical trials. This was for them to share ideas and then build capacity towards the proper and effective regulation of clinical trials in the continent. Initially, the mandate of Aurum was only for clinical trials, but later it's been expanded to include all other medical products, including vaccines, and also like doing joint reviews to get products approved. That's post clinical studies. So what's the situation of regulation of clinical trials in Sub-Saharan Africa post Aurum? As I said earlier, even a lot of countries didn't have effective laws to regulate clinical trials. So promotion of effective laws for the regulation of clinical trials, and basically now almost every country in Sub-Saharan Africa has a law, functional law in place, and ethics committees at least to see that studies that are going to be conducted on the continent are ethically, scientifically sound and ethically sound and scientifically valid. And one of the things that is done is building regulatory capacity in NRS and then ethics committee through the development of the Aurum tools for the evaluation of clinical trial applications. That's quality, that's the IMP, the investigational medical product dossier, that's the non-clinical with respect to pharmacology, toxicology, and then pharmacokinetics. And of course, there's also a template for the clinical evaluation. And there's also the development of the Aurum guide for GCP inspections. Yeah, previously, I'm sure before 2007, you do a Google search, you will find out that practically almost all the studies that were done on the continent, when there are no records to verify if they were inspected or not. But since 2007, that has changed, and now most countries use the Aurum GCP guide to conduct GCP inspections. And then recently, there's just the Aurum guide for safety monitoring in clinical trials. It was observed that yes, the studies were being approved with proper review of the protocol IB and IMPD, and inspections were being conducted as per GCP. But then after the AEs and the SAEs, that most countries were not paying attention to, so Aurum decided to tackle that by bringing this Aurum guide for safety monitoring, whereby countries would have to build capacity to monitor the safety while the trials are going on, try to assess causality and assign costs. And also there's a platform for joint review of clinical trial application and then joint monitoring of clinical trials. Yes, as you can see, most of the countries are at different maturity levels. So it's only by working together that the continent can benefit most from those who are far advanced to review clinical trial applications. And then the challenges is inadequate capacity building opportunities for regulators that exist because, yeah, and as you can know, Sub-Saharan Africa also has a challenge with language, the Anglophone, Francophone, and then the Lusophone, that's the Portuguese. Yeah, for Anglophone, there are some training opportunities, but difficult for Francophone and then the Lusophone. So that's something that actually tackled. Poor harmonization with ethics committee timeline leading to delay in approvals. Here on the continent, we've not been able to harmonize these timelines, and that's also quite a challenge, and it can affect the continent from attracting trials. Then insufficient platform for sharing ideas and communication among across NREs. And of course, lack of robust electronic submission portal for clinical trial applications in most of the countries. So what do we think will be the way forward? That's a human resource capacity building through the industrial regulatory attachments, of course, training among ourselves, whereby the much more well-resourced and well-capacitated regulatory authorities can help those that are upcoming, seminars and then annual conferences on the continent where we share ideas. Then we establish a common platform for sharing ideas, communicating and networking. Then by improving the regulatory practice among regulatory authorities, especially as with respect to timelines and then fees, and then synchronization of timelines with ethics committees and other stakeholders to prevent delays in authorization process, which is a very big disincentive to sponsors, and then development of a robust electronic submission platform. And as I might say now, under the African as AU NEPAD, they are piloting what they call that's a smart safety surveillance that will be used for both clinical trials and then post-approval products for us to enable regulators to monitor real-time safety of products that are approved on the continent. This is the end of my presentation. Thank you. I'm ready for any questions.
Thank you so much for that presentation and for taking us through some of the challenges that you see, and as well as highlighting some of the issues that really could be strengthened to try and facilitate reviews of these particular studies. I'm going to move on to introducing the second presenter, and that is Julian Nyombairi, and he is Country Director at Project San Francisco in Rwanda. And he's going to take us through a presentation on experimental vaccine trials, particularly looking at G003. Over to you, Julian.
Thank you very much, Cathy, and thanks for the opportunity to provide an overview of this experimental trial. I'll be presenting briefly on the G003 clinical trial. That is a phase 1 clinical study that is evaluating safety and immunogenicity of the EOD GT860 mRNA vaccine in HIV non-infected African adults. Next slide, please. So I'll provide a brief background and the rationale for conducting this clinical trial and provide an overview of the trial protocol and design. Next slide, please. So as a general consensus in the HIV vaccinology field, it's believed that an effective and efficacious vaccine against the virus will be one that should induce immunity against the all circulating HIV viral strains. And this is to say a vaccine that will be able to generate broadly neutralizing antibodies, BNAbs in abbreviation. So these broadly neutralizing antibodies have been isolated from chronically infected individuals as a result of the infection, but HIV vaccine candidates that were tested to date were not efficient in eliciting these neutralizing antibodies. There are in the field currently several BNAbs that are in development for HIV prevention and for cure. And specifically in this clinical trial that we're currently conducting, the immunogen is designed to trigger the host's immune system to generate HIV BNAbs of the specifically the VRC01 class. Next slide, please. So as I mentioned earlier, broadly neutralizing antibodies are a result of HIV infection, and this is due to the continuous interplay of autologous virus escape and antibody maturation. And it's approximately, from the data we have currently, 5 to 10 individuals that are HIV infected develop BNAbs after a period of approximately 3 years of infection. And HIV vaccines have not been able to replicate this strategy. Next slide, please. So the immunogen that is being tested in this clinical trial is the engineered outer domain of the HIV virus envelope that is specifically designed to target the germline into producing broadly neutralizing antibodies. So germline targeting is the process of using a vaccine or immunogen to stimulate the small number of precursor B cells that have a potential to proliferate and mature and produce broadly neutralizing antibodies. The germinal center is where B cells become activated, proliferate, and increase in affinity by somatic hypermutation. In order to achieve this specific strategy, the immunogen must try as much as possible to mimic the natural process of viral mutation, and this will be done by several boosting with different immunogens. We should use one vaccine that prime or trigger B cell precursors, and then subsequently with sequential vaccination that will polish the initial immune responses and refine them towards BNAbs along the way. So the immunogen that is tested in the GO3 clinical trial is designed to target the specific class of BNAbs that of the VRC01 precursor B cells that can develop into BNAbs that target the specific CD4 binding sites. Next slide, please. So, as I said earlier, the immunogen is an HIV protein that is engineered to mimic HIV envelope, presenting it in a way that activates the precursor cells of the germline and start them down into the path towards the eventual production of VRC01 BNAbs. And this immunogen was developed in collaboration with the Scripps Research Institute. So the engineered outer domain monomer naturally assembles into an icosahedral nanoparticle structure composed of 60 identical subunits that present themselves to the B cells. And you can see in the top right 3D representation of the immunogen. The green specific particles are the immunogen that I've just described. And specifically in this clinical trial, the delivery mechanism used is the mRNA platform that is produced by Moderna. Next slide, please. So this is the study design. The protocol aims at enrolling a sample size of 18 study participants. The investigational product will be administered at a dose of 100 microgram at 2 time points. The second administration being done 2 months after the initial one. We'll be enrolling healthy African adults aged between 18 and 50 years old that are non-infected and are not at any risk of acquiring HIV infection. And participants will be followed up for a period of 8 months post immunization. The hypothesis that we are aiming at responding at during this clinical trial is that the immunogen as a germline targeting products would be able to generate detectable VRC01 class IgG B cells in the African population. The clinical trial will be conducted at 2 different sites, Aurum Institute in South Africa and the Center for Family Health Research in Rwanda. Next slide, please. So the study being a phase 1 clinical trial, the primary objective is to evaluate the safety and tolerability of the immunogen in the target population. Secondary objective is to assess the frequency and magnitude of binding antibody responses to the immunogen. We'll also be assessing the frequency and degree of maturation of the VRC01 class IgG M and IgG B cells before and after IP administration in each participant. We would like to evaluate the induction of CD4 T cell responses as well as a result of the study regimen. And we want to evaluate the influence of human antibody gene alleles on the vaccine induction of VRC01 class B cell responses. We have an additional exploratory objective of exploring acceptability of different sampling techniques that will be employed in this clinical trial that I'll explain in the subsequent slides. Next slide, please. So this is schematic representation of study duration. We will have an initial immunization at baseline, and then a subsequent one at approximately 2 months after the initial one. We will have different sampling techniques that will be able to help us assessing different immunogenicity endpoints. In the red dots represent the standard blood draws, some of which are large PBMCs. Initially prior to immunization, and later on after the first and the second immunization. The yellow dots represent fine needle aspirates that will be collecting from the lymph nodes, axillary lymph nodes, specifically on the sides from the deltoid where the ipsilateral to the deltoid where the vaccines were administered, to be able to monitor the B cell maturations. Leukapheresis is another method that will be employed, specifically in South Africa, in order to collect a large amount of white blood cells without depleting other vital blood components. Since BNAbs precursor cells are very rare, it's often that we will need large blood draws in order to isolate the very, very limited amount of these cells. And leukapheresis is a strategy that will help us collecting this, a huge amount of white blood cell without having a clinical impact on study participants. Next slide, please. Safety being the primary objective of this clinical trial, we have different committees that will be providing oversight, specifically the sponsor team from the International Head Vaccine Initiative, as well as Moderna, the study investigators and clinicians from the respective sites in Rwanda and South Africa, as well as external medical reviewers. We will have weekly safety meetings and safety reports generated, and ad hoc meetings as needed. We have an independent safety monitoring committee that is planned to review safety data at specific time points as described in the study protocol, which is composed of 5 members, including 1 Rwandan clinician, as well as statisticians. The protocol provides Sentinel study group. The initial 3 study participants constitutes the Sentinel safety group, in a way that the initial participants that were enrolled in the clinical trial were done in a staggered manner, in a way that there was an interval of 3 days, interval between each IP administration, and a pause of approximately a month for safety data evaluation by the DSMB. And I will explain in the next slides what was the outcome of this initial DSMB meeting. And the specific safety assessment that will be performed will be evaluating local and systemic reactogenicity events up to a week post each IP administration. We'll be assessing adverse events, vaccine-induced seropositivity, pregnancy outcomes should a pregnancy event occur among study participants, intercurrent HIV infections, and outcome of infections among participants who have previously received the investigational product, as well as social impacts among study participants. Next slide, please. So this is the current status of the trial. We initiated screening in June this year, and the first enrollment was performed in July. As I explained, the initial 3 study participant Sentinel group enrollments was completed on the 5th of August, and the study paused for the DSMB meeting that was aimed at reviewing the initial safety data. The DSMB met on the 29th of August, and no safety issues were identified, and approval to resume the enrollment was granted. As of today, we have 18 participants enrolled, 10 in Rwanda, 8 in South Africa, and no issues have been identified to date. And we plan to have our last IP administration in the last volunteer sometimes mid-November this year. Next slide, please. We would like to really acknowledge the study team at CFHR, Aurum Institute, at the International AIDS Vaccine Initiative, the lab scientists at Cathy and Akanmori sites, and last but not least, the study participants. Next slide, please. We are grateful for the funders of the study who are making this work possible. Thank you very much. And I'll be happy to take any questions during the Q&A session. Thank you.
Thank you so much, Julian, for that really great overview of one of these studies and the progress that you've made right through designing it, through getting the approvals, and right through to your recruitment processes. Thank you so much. We have one final speaker, and I'd like to present Jerome Singh. I'd like to invite Jerome Singh to speak. Jerome is Adjunct Professor in the Dalla School of Public Health at the University of Toronto in Canada. Jerome, over to you.
Thanks, Cathy, and thanks very much to the organizers for inviting me to the session. And I'm going to try to maybe do a few things in the session. One is try to bring the issue of experimental medicine and how it actually relates to not just the field of clinical trials, but more specifically how it relates to the field of ethics as well. So just let me start off by giving a few introductory comments. You can move to the next slide, please. So I think, there's some key issues that I wanted to maybe highlight just to give us a background here. I think, while we currently have several effective HIV prevention modalities, including oral PrEP, the dipivurine vaginal ring, long-acting cabotegravir, we still feel that a vaccine is the most effective way to the ongoing HIV epidemic, and it's probably the best preventive solution that we're going to have. However, as I'm sure all of us know, developing a vaccine against HIV has proven extremely elusive. In the past, numerous approaches have been tried to develop an effective HIV vaccine, and they include the use of a carefully selected HIV protein, multiple in fact, specially designed antigens, and also the creation of a mosaic protein from different HIV strains. All of these methods used a single optimized antigen in the vaccine, but were shown eventually to be ineffective at eliciting broadly neutralizing antibodies. So to achieve our goal of developing an effective HIV vaccine, we basically need to design immunogens to elicit broadly neutralizing antibodies against HIV. Now, there's many possible solutions. One possible solution is to use more than one antigen in a vaccination protocol. But this raises a number of questions. How many antigens are necessary? How different should they be from each other? And in what temporal order should they be administered? And we also then need a basic understanding of the role of each antigen amino acid in the antibody-antigen interaction. Now, these are lots of questions and uncertainties. And as a result of these uncertainties, 3 steps were proposed by the HIV field to move the field forward. First, there was a proposal that we need to create a special purpose antigen to activate the correct precursor B cell. Now, since this precursor won't bind to a native HIV, as a second step, the HIV field, vaccine field proposed one or more antigens to be used as intermediaries. And in the third step, one or more antigens must be used in a mixture or a sequence to increase the breadth of the antibody population. Now, just in simple terms, because this was something I had to get my mind wrapped around as an ethicist and as a regulatory person. But essentially, if you follow the traditional clinical trial pathway to realize the strategy, it will involve a sequential research approach through multiple preclinical and phase 1 trials. And if we continue to take this approach, it'll take us at least 10 to 12 years, if not longer, to bring an HIV vaccine to the market. And that's assuming we succeed in understanding the role of each antigen amino acid in the antibody-antigen interaction. And there are several of them. Now, such a long time frame is not feasible from a scientific, from a public health, or an ethics perspective. And I think this brings us to where we are today, and that's we need to consider an alternative pathway to move the HIV vaccine field forward. And experimental medicine may offer us a way forward. Next slide, please. So I think the one thing that I wanted to do is I wanted to distinguish between the difference between experimental medicine and phase 1 trials. And we've heard some presentations, and I think what is important, most people who are involved in listening on this call have an idea, either an advanced idea or even a basic idea, if you're just starting off in the field, that to do a clinical trial, you need to go through several phases. And you have phase 1, phase 2, phase 3, phase 4. In the US, you even have microdosing trials called phase 0 trials. But the bottom line is that these are all sequential. And it takes a while to do each one, write up the results, and essentially move the field forward based on those results. And the HIV vaccine field is currently involved in several phase 1 trials. However, while phase 1 trials are similar to experimental medicine in several respects, they also differ. And I'm just going to highlight the difference by appreciating the implications of experimental medicine by just focusing on 2 aspects that distinguish the two. So in terms of the primary endpoint, a phase 1 trial has as its primary endpoint safety and tolerability. And the secondary endpoint is usually scientific in nature. On the other hand, you have experimental medicine, where the primary endpoint is scientific in nature, and the secondary endpoints are safety and tolerability. And of course, this is important here that while I'm saying that safety and tolerability is a secondary endpoint, it doesn't mean that it's taken any less seriously. But the bottom line is that, as you can see here, when you're looking at a trial and you're looking at this through a lens of an ethicist or a reviewer, or even a community advisory board member who's trying to look at this, the goals of experimental medicine and phase 1 trials are different. The second part, or the second way that they are distinguishable from each other, is their goal. So in a phase 1 trial, the ultimate goal, hopefully anyway, is to develop a product, is to develop an HIV vaccine. But with experimental medicine, the goal may not necessarily be at this point in time to develop a vaccine based on what they're testing. It may be something that is further downstream, but the bottom line is that this is a precursor to developing a product. They want to test, scientists involved in experimental medicine wants to test a hypothesis. And maybe even in the process of testing that hypothesis, generate other potential hypotheses indirectly. But we must be cautious about experimental medicine getting mired in hypothesis generation versus hypothesis testing. The bottom line is that, I think just for the purposes of an ethics discussion, the primary goal of experimental medicine must be to test hypotheses and not to generate hypotheses. In other words, you can't just do research for the sake of doing research because you're curious. You've got to have a set goal, and you must test this goal in your research. Now, I've just tried to highlight very briefly that while experimental medicine and phase 1 research appear similar, they're not. And they have many commonalities, but they also have differences. So I want to move on now to the next slide, please. Then I want to look at safety and risk mitigation. So both with phase 1 trials and with experimental medicine, safety and risk mitigation is absolutely crucial in designing the trial. And in experimental medicine trials, you'll find that you're not conducting the trial with the prime aim of benefiting those in the trial. So you find that investigators have a specific responsibilities to participants in the trial, and they have to ensure, of course, in all trials, that participants are not harmed as a consequence of taking part in the trial. So when investigators are designing an experimental medicine trial, they've got to take this into account, and safety and risk mitigation has to be absolutely paramount. The second ethics issue that I want to consider here is knowledge generation and appreciation. There are several ethicists who are sitting possibly on this call, research ethics committee members, community advisory boards, and even scientists. And some people may have understood more of what I said so far compared to other people. And I think that this speaks to the issue of knowledge and appreciation. We've got to ensure that research ethics committee members throughout all countries that could be potentially hosting experimental medicine trials, not just research ethics committee members, but community advisory board members and participants. All of these constituents need to appreciate the nature of an experimental medicine trial. You need to explain the primary goal of an experimental medicine trial in a simple way without any regulatory or scientific jargon, so that everybody's on the same page. It speaks to the need, of course, of what I'm saying, the need for capacity building for research ethics committee members and community advisory board members to sensitize both these parties and even scientists as well as to the goals of experimental medicine. Because sometimes even scientists who are knee deep in the science may not be able to see the wood from the trees. So it's important that everybody's on the same page, that when a scientist is explaining what experimental medicine is, they don't mix it up with a phase 1 trial. Or if they are doing a phase 1 trial, they don't conflate experimental medicine with phase 1 trials. They are different, and it's important that scientists, community advisory board members, and ethics reviewers, and of course, study participants are all on the same page as to the goals of experimental medicine. Next slide, please. Another ethics issue that's important to look at here is, of course, the issue of value and social value. So you'll have to ask, before you do an experimental medicine trial in a country, for example, in Africa or Asia, does this actually have any sort of benefit? And does this have any sort of social value? So social value typically speaks to the issue of, will this have relevance to the host community? Will this trial actually be valuable to them? But I think, it's not just looking at social value, we also have to explore the broader notion of value. And when we're looking traditionally, and I'm talking here to ethicists who review studies, traditionally, when we do research ethics review, we conduct a risk-benefit analysis. And I think the important issue to bear in mind is with a experimental medicine trial, there's no benefit that accrues to the trial participant. So when you're doing a risk-benefit analysis here, immediately you're going to move in the direction of more risk or more potential risk, but no benefit. So I think when we're looking at this as a reviewer, reviewers need to take into account the notion of not just risk-benefit, but the broader notion of value. So you'll find here that when we're looking at benefit and value, there's broader notions of this here. We're not just looking at individual benefits. Personal benefit versus generating knowledge to aid the field is acting in the greater interests of the wider community. And there's sometimes a conflict between what we perceive to be what's in the best interest of those who participate in a trial and what's in the best interest for a future population that may derive benefit from the intervention later downstream. So I think that research ethics committee members and even community advisory board members, when they're looking at experimental medicine trials, they need to remember that it's not just risk benefit, it's also value. And we have to look at what is the value of the trial, what hypotheses are they testing, how is this going to move the field forward, and is this valuable to move the field forward? And these are the important issues to consider as both community advisory board members, research ethics committee members, and even in terms of messaging when we're looking at the role of scientists in explaining what experimental medicine is. We also have to explore from an ethics perspective the notion of reciprocity. And that means that if this host community and study participants are going to help to answer these very fundamental but very early stage questions, and this is going to help to move the field forward, will the end product be made accessible to the community when this vaccine is commercialized further downstream? Because we've seen this happen, of course, with COVID vaccine trials, where you found African communities, for example, in South Africa, were hosting COVID-19 vaccine trials, but they did not get access to the vaccine at the same time as Europe and North America. It was months and months later, and we sat in the queue like the rest of the world. And hosting the trials made had absolutely no benefit for the host community. There was no additional value for the host community in terms of getting prioritized access. So when we're looking at experimental medicine and we're answering formative questions that is going to move the science forward, we've got to ask ourselves the reciprocity question as well. We're making a sacrifice by involving ourselves in this trial. We're putting ourselves in line, and we're helping to generate these important answers to your questions. But where does it leave us? Yes, this has social value in our country, but will we get some sort of prioritized access for helping to answer this question? And this needs to be put on the table quite early on. And the last question, or the last point that I wanted to raise, is the payment of incentives. Another important ethics issue in experimental medicine trials. Should you offer incentives? Well, you know, the provision of incentives in clinical trials is well established, and it's considered ethically acceptable. However, we do need to take into account several different issues. You know, while it can motivate participants and it can show it's a way of reflecting respect for the participants, I think it's a matter of whether the incentive is an undue inducement or not. So in other words, the nature of the incentive must be taken into account. Are you offering money? How much of money are you offering to participate in this in this experimental medicine trial? And you'll find that this is where community advisory boards have to come in, because they have got to determine the appropriateness of an incentive, because an incentive in the US may be an undue inducement in an African country, and the payment of an amount in one country may be hugely inappropriate in another country. So I think this is an important role for community advisory board members to play, is to determine that if we're paying an incentive to recruit people onto an experimental medicine trial, what should that amount be and what is appropriate in the local context. So the last slide that I want to look at now, so you can move to the final slide, is that, you know, in conclusion, you know, the traditional research pathways that we have followed for decades and decades in biomedical science have not yielded an HIV vaccine. It's partly due to the nature of the HIV virus, but the bottom line is that it's not, the traditional pathways haven't yielded a vaccine. So to continue to follow a traditional pathway to develop a vaccine is not feasible. It's not feasible scientifically. It's not feasible if you want to confront and deal with HIV from a public health perspective, and neither is it acceptable from an ethics perspective to wait another decade or and a half for an HIV vaccine. So experimental medicine could offer an expedited pathway to developing an HIV vaccine. But again, just in conclusion, safety considerations are paramount. Clear communication on the part of scientists, regulators, the ethics community, and community advisory boards are important. And this all speaks to capacity building. And in other words, we need to take how and what experimental medicine is and train our regulators, our research ethics committee members, and our community advisory board. And we need to communicate these notions in a very simple way in informed consent documentation, because all of these things are critical to building trust and confidence in the field of evidence medicine, experimental medicine. And I'll stop there, but thank you very much for your attention, and I'll be happy to take questions in the session.
Thank you so much, Jerome, just for providing us with those insights about what lies ahead and how these experimental trials need to really be messaged, particularly understanding of the purpose of these trials and how to do slightly different risk-benefits assessments. That was really helpful. At this point, I'd like to introduce 3 additional panel members and then open it up to a panel discussion. And I think we're making fairly good time, so hopefully we'll get some discussion going. The first panel member I'd like to introduce is Bartholomew Dicky Akanmori, who is Regional Advisor for Vaccine Research and Regulation in the WHO Regional Office for Africa in Brazzaville, Congo. Welcome, Dicky, as well as Gavin Churchyard, who is CEO of the Aurum Institute in South Africa, and Michael Makanga, Executive Director of EDCTP. So we're really excited to have these 3 experts on our panel, as well as our 3 presenters. So I'm going to be directing some questions to our presenters, but in the meantime, please feel free to use the Q&A, not the chat, just the Q&A to post any of your suggestions, comments, reactions, etc. I was also asked by the organisers just to kick off the panel discussion by just reminding everyone of the definition that was put forward by Robin Shattock for experimental medicine trials, and that's a clinical investigation undertaken to test or generate a scientific hypothesis that advances vaccine discovery and development, but where that intervention will provide no direct prophylactic or therapeutic benefit to participants. So I'd like to just start off by first just directing some questions around the regulatory issues, and beginning perhaps with Dicky. Obviously, experimental medicine is not a new concept, but as Jerome has highlighted, it's become very heavily talked about now in the context of HIV vaccine development. Just from a regulatory perspective, in your view, is this clinical study being considered differently by regulators, for example, from other first-in-man studies or phase 1 trials? What has been your experience with the regulators when faced with these particular studies? If you could just let us know some of your thoughts on that, please.
Well, thank you very much. And we have had 3 brilliant speakers who have clearly outlined, from a regulatory point of view, what clinical trials are, what they entail, particularly in our settings here in low and middle income countries. We've also had a clear presentation which has defined for us what experimental medicine is. And but let me start by saying, indeed, it's not new, because let me fundamentally step back and say that once you make that transition, that decision to transition from the laboratory or from laboratory research, animal or animal experimentation to humans, you are actually embarking on experimentation in humans. And I think all those in ethics and all regulators agree fundamentally with this. And I think this then, of course, requires that you ensure that you meet certain clear criteria. And I think the first thing I want to point out is that you need to accumulate sufficient evidence that, yes, indeed, what you're doing is scientific. Now, it doesn't matter whether this product is going to go forward or it is a deliberate attempt to actually eliminate the product, because sometimes you need to do that in research settings. In product development, you need to intentionally generate the evidence which will eliminate the product, and thereby preventing people from putting resources into something which will ultimately not lead to a product that can be made available. And there are several examples of this in history. We've seen several attempts early on to use a single malaria antigen to try and develop a vaccine against a very complex pathogen, a protozoan parasite with multiple life cycles, with evolving antigenic presentation to the immune system. And, to some extent, these were experimental, these were examples of experimental medicine that helped to eliminate some products. And now eventually we do have one product, and there are several in the pipeline, which have demonstrated that you need to go beyond just one antigen. That one antigen will not work. So yes, from the regulatory and ethics point of view, this is something which has been around for a while. But as has been said, you need to ensure that this is hypothesis driven. The science is right. I'll give you an example of one that took place where the African Vaccine Regulatory Forum, AVRF, a network of regulators and ethics committees on the African continent, was approached by the HIV research community several years ago. And they were required to prepare to see if they would be willing to approve a clinical trial of an experimental vaccine against HIV, and whether they were willing to do this not as a product intended to go beyond, the clinical trial phase 1 and into marketing, to the market. And the question they asked was simply this: we do not have a correlate of immune protection, which fundamentally is one of the huge problems that we face with developing vaccines against HIV/AIDS. What we would like to know from you is, if we were to present to you a study intended to help us develop assays that can measure some level of immunity, something we can consistently measure and track and get consistently the same results and standardize across, so it can be used as a basis for comparing different antigens or candidate HIV vaccines for clinical trials. Would you be willing to consider to review this? And indeed, the regulators of the continent, particularly the more advanced countries, said, yes, why not? Because they understood the importance of having experimental medicine. So, I mean, in short, this is something which is not new and is, even within the settings of LMICs, has been considered, because the ultimate goal of ethics committees, regulators, is to have a product and to contribute to access universal health coverage and to improve health outcomes for the continent. Thank you.
Thanks so much. Thanks very much, Dicky. And it certainly sounds like the way in which the purpose of the trial is messaged and communicated by the applicant also goes a long way for how it's received by the regulatory authority. I'd just like to raise one question about AVAREF, and it's also come through a little on the chat. And I'd like to perhaps direct this to Michael. There's several Pan-African networks that can support applicants, sponsors and researchers who are interested in conducting this kind of research, and AVAREF is one of them. How could AVAREF best help investigators and sponsors with these studies? How could it perhaps facilitate or accelerate these types of studies in Africa? Could I ask Michael perhaps whether you'd be willing to address that?
Thank you very much, Cathy, for this question, and greetings to all participants. I'll be willing to respond to this, but it will be more appropriate to first hear this from the AVAREF's point of view, and Dicky represents AVAREF. Then I can maybe give the funder's perspective to this, if that is all right.
That would be perfect. That would be perfect. Please, if we could switch back then, Dicky, if you could respond to that.
Yeah, many thanks. And I think this is a very important point, a very important question, Cathy, that you highlight. And I think it's one that people consistently ask and, have in their minds, because having heard what Eric, clearly explained, the need for AVAREF and how AVAREF has evolved to alter the regulatory and ethics landscape, that indeed it represents the continent and it represents the effort of lower middle income countries to take on their own, in their own way, a very major responsibility of supporting product development, ensuring that they meet the required standards for both ethics and regulatory reviews and approvals, while at the same time recognizing that this is the most deprived, these are the most deprived countries when it comes to health, when it comes to addressing their priorities, because as you know, most of the resources in this world are applied to health problems that affect the developed countries. And in fact, there is a huge imbalance when you look at R&D investments in developing country settings. So this makes it appropriate. And indeed, yes, AVAREF has put in place deliberately and systematically some of the major steps that can help address this. One, I think, which most people do not recognize clearly, is that you need to have a platform that brings together ethics reviews and the scientific and regulatory review. You need to address these comprehensively together. And it's when you structure and bring these two together, not necessarily overlapping or taking, ensuring that or trying to interfere with the roles and responsibilities. But if you bring them together, you get a more efficient review. You also get a high quality review, because when you bring countries together, they actually share the limited capacity which is within the continent. And this ensures high quality reviews. And not just that, as has been earlier explained by Eric, it also rapidly builds capacity. And we are in settings where, global health is pushing for shorter timelines for product development. Michael knows this because he funds research. And secondly, not just shorter timelines, but they also want better products. And yet we're left with the most difficult products to handle. And now this then poses a challenge. And in developing country settings too, that where these products are required, it thus requires that you get countries collaborating and working together. And I think this is one major value of bringing these two components together to work together and ensure that they meet the timelines. And I think the disparities and the differences in submissions and processes have been such a huge challenge on the continent that they have literally discouraged product developers from attempting to work with sites to develop products. But AVRF has come to show that you can bring about convergence. You can bring about approaches which bring countries closer, develop templates, develop guidelines, develop submission packages that are very similar without any huge differences, enabling a sponsor to submit right across the continent, and particularly where case numbers are few, and also where you want to look at seasonal transmission and the effect of your intervention, then you can recruit across sites, and multi-country clinical trials then become very easy to conduct. And these are some of the values. And I think apart from this, AVRF has also brought the partners together, because I think collectively the partners can make a bigger impression. They can make a bigger impact when they work collectively in supporting regulatory and ethics systems and building capacity together. And I'll pause here. I'm sure Michael will have a lot to add to this. Thank you.
Thank you so much for those insights. Much appreciated. Michael, I wonder if I could circle back to you and ask for any additional comments. And also, please, if you could maybe describe some of the work of EDCTP. I know they're investing in capacity building, and perhaps you could also make some comments just for the audience just about that work in this regard. Thank you.
Thank you very much. Very briefly, in relation to this question, our approach is three-pronged. It is a three-pronged approach. One is from the investigator's point of view. 84% of our funding goes towards research and development. And in this case, we encourage early engagement of the researchers with the ethics and the regulatory authorities, making sure that they share their protocols early. They share the information about the studies early. As we've just had, we support both the phased research activities, but also aspects of experimental medicines that coming in with a different approach and different designs, many of which are now adaptive in nature. Secondly, we have engagement directly with the national regulatory authorities and the national ethics committees as well, providing funding available to them to strengthen their capacity to operate more independently and to address specific gaps. The third is our interaction with the convening bodies, and in this case, AVRF. In fact, we worked initially together with WHO in the setup of AVRF, and we are glad to see that the AVRF initiative has evolved over the years. Now, moving on to our capacity development approach, we have a very integrated approach in all our clinical research grants, and this spans development of the human capital through short and long-term training, as well as the infrastructure that goes with it to ensure high-quality clinical studies. What is key also is the investment in the people, and this is through a very large and comprehensive fellowship program that is at all levels. And this also includes ethicists and regulatory authorities. And one aspect to this is ensuring that you have individuals that are well-trained with the requisite expertise, but globally connected to be able to tap into resources in other regions and countries and to draw in that expertise to apply it locally. The other bit is paying special attention to the commitment to equity. And in this case, we have another approach of funding through regional networks of excellence based on regional economic communities or that structure, so that we address areas that may have special gaps that otherwise may be less competitive. We've also brought in addressing the gender gap, where we have ring-fenced funding that, for example, goes towards female researchers, and also establishment of tandem schemes where we pair up well-established individuals and institutions with those that are upcoming. So this is the nutshell part of our approach in supporting capacity development. Over.
Thank you so much. Thank you, Michael, for describing really what is a very enabling environment for these kinds of studies. At this point, I'd like to switch to Gavin, and just, Gavin, I wonder if we could ask you, just in your view, looking at other disease areas like malaria or even COVID, whether there are lessons that we can learn from these other disease areas when we're trying to think about these particular kinds of studies. Is there anything that we could draw on to make these less daunting and to use these particular previous lessons moving forward? I wondered if you could share some of your thoughts on that.
Thanks very much. So there are a number of issues that we need to consider from a site perspective, but let me just start with from the other disease entities. From our own experience, we have done an investigator-initiated and led experimental medicine trial for TB host-directed therapy, so not a vaccine, but nevertheless, it was designed as an experimental medicine done in South Africa. And so many of the ethical and regulatory challenges that were articulated, we had to address in implementing this trial. And so some of the issues that we needed to address, so just to mention, sorry, in terms of COVID vaccines, we didn't do experimental medicine trials. We did the large late stage trials. But in terms of the experimental medicine trials, so Jerome has already spoken about some of the challenges around consenting and being very clear on the lack of benefit, that the incentive structure may need to be different. One of the issues around experimental medicine trials is that they're typically much smaller than other trials, although they may be similar to first-in-human trials. And this has important implications for if there are a series of experimental medicine trials being designed to develop this sort of iterative process of immunogen design for vaccines, sites are going to have to become accustomed to doing and dealing with multiple small trials on an ongoing basis. That means doing lots of regulatory and ethics submissions, and if there are protocol amendments, etc. So it may be quite taxing for the sites. That sites are going to have to target a different population from what they are typically used to. It's more healthy and low-risk populations to deal with vulnerable groups, and those with low education levels may be a challenge in this aspect. And then these experimental medicine trials often tend to have this very slow initial enrollment. Jerome highlighted the importance of getting the CAB on board to provide input and to help with providing community education and minimizing the potential for vaccine hesitancy to arise. Safety is a very big issue, as again Jerome highlighted, and that's from when we were doing our trial, we had to make sure we were ensuring access to very high quality healthcare to manage any product-related AEs. We ensured, for instance, that we had emergency services on site when administering products, and that we had access to private hospitals and ICUs should there be a product-related adverse event. And because the clinical trial insurance may have limited cover, we have also in our trials made provision for supplementary funding if that was required to address these safety issues or care for safety issues. The other challenge that we find with experimental medicine is that the time required to complete the trial is much shorter, so you've got to really be very efficient in getting the trial done, which sites may find challenging. And this, in order to do this, you really need a very dedicated, focused staff to work on these trials. And typically what we do in sites for doing the late stage development is we have pluripotential sites, we cross-train staff on multiple different protocols, where with these protocols, you really need people very focused on that trial to make sure they get done properly and safely and on time. There's also a number of laboratory issues. So they may involve novel assays that are required and shipping to different labs around the world. And then from a pharmacy perspective, the stability data on the products being used in experimental medicine designs may not be as long as what is would normally occur for a product that is in a pathway to licensure. And so, any overruns in the time to do the trials may result in that product expiring, which would be a problem. So I think there are numerous challenges, but all of them, none of these are insurmountable, and sites can absolutely deal with this if given the required support.
Gavin, thanks so much. Thanks for taking us through the particular skill sets that are required to really host these studies comfortably, and particularly bringing in the community expertise as well. I wonder if I could now just ask Jerome, just circle back to you, Jerome. You mentioned several of the issues that are likely to be important in these studies. I wonder whether you could think about what it is exactly that applicants who are devising these protocols, drafting these protocols, how could they do that in a way that facilitated the quality review by the time it got to the ethics committee, to prevent a back and forth or to and fro? What could they do to really position these trials, in fact, do a lot of the ethics work by the time it gets to the ethics committee? I wondered if you would just revisit some of the ideas from your talk and maybe just take us through your advice for applicants devising protocols that they know are going to go to ethics.
So I think maybe what I'll do is I'll start off maybe one step back and maybe tackle a question that was asked in the chat as well, which I'll tie up with this question that you've asked. And that is, I think one of the ways to facilitate the process, and so that we are not reinventing the wheel each time, is to have a platform. And these are platform trials, or to make the experimental medicine a platform trial. And that basically means that you have a master protocol, and every time that there's a slight variation in terms of what you want to test, that actually is changed in that aspect of the protocol, but essentially the protocol stays virtually the same. And that means that when you're doing a platform trial, so for example, we have a platform trial for the WHO's COVID-19 vaccine trial. So the Solidarity Vaccine Trial is a platform trial. We have one protocol. I serve on that committee that reviewed that trial. And we have one protocol for multiple interventions that came through. And essentially, we don't have to review the protocol every time a new vaccine is added to the protocol. And similarly, you'll find that there have been several other platform trials that were done during COVID-19. So I think the HIV vaccine field needs to move into platform trials. That means that the master protocol needs to be quite clear. And the central messaging needs to then be, of course, taken up and adopted in all the other subsequent trials. But coming back to what you are saying now, is that if you do a good job the first time round, you don't have to keep on reinventing or finding a way to explain something. So essentially, coming back to what I'm just talking straight out of my head and I'm not giving ideas here, but I'm using this as a very simple example where, the HIV vaccine field is extremely complex. And when you're looking at trying to find the right protein to add to an ingredient for a vaccine, as an example, it's quite a difficult concept to explain to a layperson. And, I think the challenge here is to find a committee like a lexicon committee that puts together words and ideas in a very simple way. So scientists, regulators, and ethicists love complex terms. We love them. We love the jargon. But of course, this doesn't mean anything to a person on the ground. So when I review a study and I'm looking at an informed consent form, I often have to send it back and tell them, what does this even mean? You're using scientific jargon. Nobody knows what this means. What does randomization mean? You are assuming that somebody knows what you're talking about. So I think, you have to really take this back. And I think this is the tension sometimes there, where sponsors, ethics committee, review committee may require or mandate the inclusion of certain terms. And this is problematic when you're taking these same terms and exporting them to a research setting that may not use these terms, or where this culture or legalese culture may not be something that is acceptable. So if you're trying to explain that we're trying to find out whether adding this is going to make the vaccine better, a very simple analogy would be to say, we're using this, it's like an ingredient to making food. We're trying to find out what's the best way of making it effective, something like that. But the bottom line is that there's often a disconnect between explaining something in a very simple way and also a regulator or an ethics committee, who are staffed generally by very highly educated people, thinking this is too dumbed down, this is too simplified, and you need to put all these elements in because we require risk to be mentioned, the word benefit to be mentioned. We require all these different terms, but they're actually following a separate paradigm. And those types of paradigms are not going to be necessarily understandable for somebody who's a layperson. And outside of the field of HIV vaccines, COVID-19 vaccines are lay people when it comes to HIV treatment protocols. The term layperson here is somebody who's just not familiar with the exact terminology or the methodology of a particular field. So I think, when we're designing these forms or documents and our protocols, we need to make the terminology very accessible, not only for study participants, but also for reviewers at the research ethics committee level and also for regulators as well. And I think it also requires a mindset change on the part of sponsors, where they need to really unthink that the way that they impose, regulations on their sponsoring countries. So, there's a bit of give and take here. And I think we've got to leave it up to local ethics committees, community advisory boards to read these documents. And if they say this is not understandable to them as a representative of the community, that says a lot. We need to reset and redo this properly again. So I think, we've learned lots of lessons from HIV. Those lessons have now been exported to other fields. And the way that informed consent forms are being done for other fields now have taken several pages out of the book of HIV. And I think similarly here, you'll find that we really need to simplify the terminology, and we've got to use terminology that the equivalent of a 12-year-old would understand. That's the general rule of thumb in research ethics, is design your informed consent form and study documentation for the level of a 12-year-old. If a 12-year-old, literate 12-year-old can't understand this, you've got a problem. So, this is just one way of looking at this. And I think that depending on who you involve in a study, we need to remember there are important social, cultural factors that can impact on how language is used, how language is understood, and the way that phrasing should be done. So I think when you're designing an informed consent form, we often talk about ethics, but we also need to take into account culture, and social factors as well. And that means that we've got to find culturally appropriate terms and culturally sensitive terms to use in our forms that we put together. And regulators and ethics committees need to be aware that this is being done to sensitize the local population, and that while you may have one protocol that's been formulated, in a sense, a master protocol, the way that it's being translated and the way that it's going to be done at the, at the site level might be slightly different. And this is something that has to be accepted. So I think, there's a mindset change. This is not something new that I'm saying. It's quite well established in the literature, and all fossil review protocols know this is an old age problem, is trying to put something in a very simple way. So I think sometimes when protocols are being written by investigators, they do this as if they are writing it for a funder and impressing them with big terms and complex terminology, but that's not who your audience is. You need to undo that terminology. Your audience now are regulators and ethics committee members who may not be familiar with that science or that methodology, or even what we're talking about, your experimental medicine. They may assume that they know phase 1 to phase 4 clinical trials and the basics, but that's not what experimental medicine is. So, I think at the end of the day, you've got to tailor-make your forms to a way that is going to be responsive to the study that you're doing. I've spoken a lot, but I'll maybe stop there.
Thank you so much, Jerome. In the remaining minutes, I'd just like to direct a question to Julian, one of my own, and then also one has come in from the floor. Julian, you described the G003 study. Could we just ask you for your thoughts on how the study was received by the regulators and the research ethics committees, whether you had any particular concerns from those two bodies about the study, just in your experience? And then from the floor, someone has asked, may the phase 1 trial include questions to users on their views of uptake of the vaccine when it's available? And has the sponsor discussed access to the vaccine if it's proven effective? And is the pathway discussed with researchers? I know that's quite a lot to give you, but I wondered if we could just have some of your thoughts on those. Please go ahead.
Thank you very much, Cathy. I'll try and be brief. So yeah, the submission of these protocols have generated a lot of interest from our regulatory bodies, both the national ethics committees as well as the FDA. So the main reason being the novelty of this experimental trial. We are using the germline targeting is not very used for our regulators. They don't see often these protocols using vaccine using this strategy. And in addition, we are using mRNA platform as a delivery method, which for even for most of us scientists in the field have heard this for the first time during the pandemic with the COVID vaccine. So this generates a lot of interest, and with almost all the regulators in country, we've had requests for additional data, additional safety data from preclinical studies, as well as very early phase 1 studies, as well as stability studies for the investigational products themselves. So although this may represent an obstacle, especially when you have very tight timelines for study initiation, I personally see this as a capacity building opportunity. This concept in vaccinology being relatively novel, it's an opportunity for the regulatory, when they receive this additional data regarding the IP and the studies themselves, to increase their awareness and knowledge of these products, especially that usually the iterative character of these clinical trials may generate data that warrants future trials. So by receiving additional data, it makes future submissions or future protocols using similar vaccinology strategies much easier in the future. And other panelists have highlighted the need for capacity buildings, strategies of our regulatory bodies. Prof. Karikari mentioned seminars, and these are highly needed in the field. To the second question, about future access. I think specifically talking about the GO3 clinical trial, these are very early stages. We're trying to prove a concept. Initial study have proven that it was working in American population. We are trying to see if it's working in African population as well. But in order to have a clear picture of when and where this will lead to an efficacious vaccines that will lead to licensure of the vaccines for broad use, we are not yet there. But issues of access are already being discussed, and we are trying to assess this. While the studies are ongoing, these are activities that the sponsor team as well as ourselves in our context, national context, are seeing, how this will be feasible in the years ahead once we have a product in the field. And was there a third question? I can't really remember.
No, you handled it. Thank you so much, Julian. I appreciate it.
Thank you very much.
There's just 2 minutes left, and I wondered whether at this point I should direct a question to Eric, or whether we should try and close given everybody's time commitments. So can I just please reach out to Elena and just see what would be the most appropriate thing at this point, please, just given the time. We're 2 minutes off the close.
I think
Elena?
wrap up, getting maybe 30 seconds from each one to summarize or giving key message would be nice. Thank you.
Right. So I'd also just wanted to say, just thank you to everyone for just giving us a better sense of these studies and the way in which we need to intervene with participants, research ethics committees, regulatory authorities, CAD members, and even at the site level. Could I ask all of you just to perhaps make 1 or 2 closing remarks? Would that be acceptable? Just top line messages before we close?
Can I start? Cathy, can I start?
Yes, go ahead, Eric.
A good forum, but then there's one thing I want to say. When regulators demand for additional documentation, I mean, with respect to best regulatory practice, if the information that you are requesting will change your decision, you would have to demand for it. So mostly documents are not demanded because you need to have them on record. I mean, you need that information to take the right decision. Like I wanted to ask Julian that you said you've done phase 1. Where did you do the first in human? If the first in human has been done in a stringent regulatory authority that we can rely on, yes, then maybe you can waive the non-clinical data and so on. But if you don't have that data, I would make sure I see all those documents before I request I might recommend for the study to proceed. So that's it.
Absolutely.
We still use the principle of reliance. Yeah.
Right.
Absolutely.
Yes.
Thank you very much.
I see a second webinar. Yes, I see a second webinar with some of these nuances. Thank you so much, Eric. Would anyone else like to make just a quick closing remark before we go?
Yeah, I can. So it's wonderful to have these discussions. It's absolutely appropriate that for HIV, a high priority disease for Africa, that African researchers, ethics committees, and regulators are part of this process. We don't want to be left behind. And in fact, African investigators should play a leadership role in thinking through these big issues and helping in the design of these trials so that we're not just passive recipients, that we are active participants. We have the ability and we're ready and waiting to play a leadership role.
Thank you.
No, I mean, I couldn't agree more with you on this. And I would say, I couldn't agree more with you on this. And I'll even go on to say that, yes, African investigators should take the lead. And I'm glad that we have someone from Rwanda who is leading this investigation. But I think I just want to end on the note of access, because I think we cannot, overemphasize this point. I mean, we've seen what happened with COVID-19. We still have inequity, and sadly, the LMICs, particularly our continent, is often the continent that's left behind. So I think early on in product development, access must absolutely be brought to the fore, which is why the African CDC has come up with a target for us to expand vaccine manufacturing on our continent, and Rwanda is one of the countries taking the lead. And I would love to see if there's success with this product, that eventually Rwanda does manufacture this vaccine, regardless of the fact it's mRNA. In fact, they are even targeting mRNA anyway, that they manufacture this vaccine and we have access. Thank you.
Thank you so much, Dicky. We are 2 minutes over time, but I'm sure we've got 30 seconds just for any more closing comments. Would anyone else like to make a closing comment?
Thank you very much, Cathy. I just want to add that this is an area on experimental medicine research that really needs to be sensitized better, so that there is more cooperation and a common understanding on the side of the regulators, the policymakers, the community, and also the risk and the funding agencies. It is really important. And local leadership in these studies is critical and has to be supported. Over.
Thank you. Thank you so much. Jerome, would you like to make a closing comment? Just before.
So only
a few seconds.
I think probably the most important thing is, if you're a research ethicist or if you're a regulator, even if you're a community advisory board member, I think we need to get our minds wrapped around what experimental medicine is. It's not the same as a phase 1 trial. And I think that when we use the term, all research is experimental, but when we're talking about experimental medicine in the context of what we were aiming to do today, it's a slightly different topic. So it means all of us have to take our responsibility of learning about the subject matter, and also those who support these types of initiatives have to also try to get involved in the training of people, so that we're all on the same page when we're assessing risks, benefit, value, etc.
Great. Thank you so much. Much appreciated. And Julian, I think it's just over to you for closing remarks.
Yeah, thank you, Cathy. So, I mean, the vaccinology field is evolving very fast. This is not only for HIV, for it's the case for COVID, even in oncology. So there's and from the different talks we've had during this webinar, with a common comment that has been coming up is the need for capacity building, and we need to keep the discussion ongoing on how to make this happen. Whether we would need seminars, international or within country, that may generate some capacity building in anticipation of upcoming trials, not only when we have new protocols coming up for submission, but in anticipation of the study that we have ahead. We could sit with regulators, explain what we have in the pipeline, and it really working one step ahead, planning for successes in submissions, but also in study implementation. So I hope this talk will be, will keep on going, and that will bear fruits into generating capacity for our regulatory bodies, national and continent-wide. Thank you very much.
Thank you. Thank you so much, Julian. It's been a pleasure to hear from all of you. Elaine, I know we're a little bit over time, 5 minutes to be precise. I don't think that's too bad, and I'm going to hand back to you. Thanks very much.
Thanks, Cathy. No problem. I think it was worth it to summarize and wrap. Thanks to everyone, to the speakers, the panelists, and to you, Cathy, for your time today. Thanks to the participants for also showing your interest in this relevant topic. You will get a recorded session in the following days by email. Please fill in the survey that you will get at the end of when we close the session, so that help us to improve for future, even to get ideas for future sessions. And having said that, I think we can close here. And thanks so much. Have a nice rest of the day.
Thank you so much. Thanks to the organizers. Bye.
Thank you. Bye.
Cheers, Elena.
Thank you. Bye.
Bye. Bye, everyone.